All right, bio buddies. We're going to be starting our unit on genetics, and we're going to be giving you some really important information to start things off, so hang on to your seats. Here it comes. Hey, not a big secret that organisms pass on their traits from parent to offspring. We've known this for thousands and thousands of years, and when we're looking at the branch of biology known as genetics, it's basically one of the oldest applied branches of biology because we've been doing genetics experiments for centuries, trying to develop the best plant varieties for food, trying to go ahead and get the best, strongest, and most useful stock animals, even getting the ultimate little puppy dogs. And even though we've been doing this for, like I say, thousands of years, it wasn't until the last few hundred years that we started to unravel the reasons why genetics actually works and some of the patterns and processes involved. Well, one of the guys that really put us on the right path was a Catholic monk living in Austria, not Australia, but Austria, named Gregor Mendel. He was doing most of his work in the mid-1800s. And, he might look like this mild-mannered milquetoast of a monk, but this guy was a veritable giant among biologists. He was a genius, a certifiable genius in every sense of the word. And his experiments, like I say, laid the groundwork to our understanding of modern genetics. Now, a lot of the textbooks go ahead and portray Mendel as this little guy who kind of sort of puttered around the garden and played with peas in his spare time. But monasteries back in that time were kind of self-sustaining institutions, and a lot of the food they grew their own. And Mendel's garden probably looked a lot like this than your backyard garden. And he chose pea plants as his organism of study. He got kind of lucky because they had a lot of contrasting traits that he could find in two different forms. They were easily bred. All you had to do was take a little bit of pollen from one flower and bring it to another, and lo and behold, you could go ahead and breed these puppies. Kind of sort of had to wait for his results, but he had generations and generations upon generations of data to go ahead and analyze. We want to look at one of his experiments here, and this is a classic one. Before we begin, we have to go ahead and introduce a couple of terms that breeders oftentimes use: purebred versus hybrid. And we also want to go ahead and link these terms up with their biological counterparts. Purebred is the same as homozygous. Homo is same, right? Zygous sounds a lot like zygote. So imagine a zygote which has received the same genetic information from both parents. That's what a purebred is all about. A hybrid, by contrast, is going to be an organism which has received different genetic information from each parent. It's heterozygous. The zygote got different information, hetero means different, from each parent. Well, in one of Mendel's classic early experiments, he took a purebred or homozygous tall plant and cross-pollinated that with a homozygous or pure-breeding dwarf plant. Now, he might have expected a medium. Hey, purebred tall versus purebred short. It would make sense to get a medium. Instead, everybody from that first generation turned out tall. Now, I know that you're probably thinking that, hey, there's probably something going on with dominance and recessiveness. I know you've heard of dominant and recessive. Well, dominant is going to be the form of the gene or the trait which is going to be expressed in the hybrid. The recessive one is the one that's hidden. We know that in that hybrid, you must have got some dwarf, some short information from one of the parents. Yet, it didn't show up. Now, the interesting thing is, when he crossed two of these hybrid individuals from that first generation, he ended up with both tall and dwarf, but always in a particular ratio. He always ended up with about 75% tall and 25% dwarf. Now, from analysis of experiments like this, Mendel was able to go ahead and develop some important principles which showed us how genes actually work. More about this in a sec. Now, you might not really seem like a super genius yourself, but you already know more about certain aspects of biology than Mendel ever did. So we're going to fast forward and add a couple of terms here that Mendel didn't necessarily use, but it's going to make our understanding a little bit better. Now, don't forget purebred, homozygous, hybrid, heterozygous. Need those terms. If you don't have those terms, you might as well jump in front of a moving train like right now. Commit those to memory. One thing you already know about are the term genes. Now, Mendel never used the term gene. He used the term hereditary factors. But you already know that a gene is just a location on a chromosome, and that codes for a particular trait. You also know that genes are made up of DNA, and genes are nothing more than a sequence of nucleotides on that DNA molecule that's going to code for a particular protein. Make a protein at a particular time, you're going to get a particular trait expressed. Well, normally when we talk about genes, we can talk about different forms of a gene. For instance, we were just looking at the gene for plant height, and we could have a tall genetic information or short genetic information. So we can go ahead and refer to this as an allele, a tall allele versus a short allele. Different forms for plant, or different forms of a gene for plant height. You can have the same thing for flower color. And you can have a white allele or white information in purple information. The white allele or the violet allele. Or seed color, yellow seeds versus green seeds. The yellow allele versus the green allele. Now these terms are going to go ahead and help us describe genotype. You might have recalled from our earlier unit in nucleic acids, genotype is just the type of genes you have. Well, in our case, we're going to rename that and look at the type of alleles that an organism carries. And up until this point, if we wanted to write a genotype, we'd have to write all those base pairs that would actually go ahead and code for a particular protein. That's ridiculous. We don't want to write all that stuff. So we're going to have a kind of a summary here. We're going to go ahead and write a genotype as just a couple of letter combinations. And if we're looking at, say, plant height, we can use the letter H to determine plant height. But note we can write the letter H as a capital or as a lowercase. If we write it as a capital, well, that would denote our dominant allele. In this case, our dominant allele gave us a tall plant. Lowercase, hey, we can go ahead and use that as our dwarf allele, or recessive allele. So, whoops, that's supposed to say short. Dude, I didn't write a bad word. That's actually short. It's short. It's short, dude. It's short, just like me. Dang it. Anyway, a genotype for a homozygous tall plant, big H, big H. A genotype for a homozygous short plant, little h, little h. A genotype for a hybrid individual, big H, little h. Now, when writing the gene pairs, if there is a capital letter, always put the capital letter first. All right? You should practice with this, but remember when you write genotypes, you're always going to have two letters, and you're always going to put the capital letter first. Now, phenotype, hey, phenotype is just what the organism looks like. Is it tall? Is it short? Does it have white flowers? Does it have purple flowers? Does it have green seeds? Does it have yellow seeds? And so on and so forth. So let's apply this to Mendel's experiment here. If we wanted to go ahead and write the genotypes of the parent plants, the original parent plants, remember we had a purebred or homozygous tall. So, big H, big H. And we wanted to cross that with a homozygous recessive or a homozygous dwarf or a purebred short plant, little h, little h. Note the parents are both going to have two alleles for each trait. Well, the offspring here, you're going to get one allele from this parent and one allele from this parent. And your hybrid individual is going to have a big H, little h. It's going to be heterozygous. And no matter which pea plant you look at from that first generation, they're all going to be hybrids. But when we go ahead and cross individuals of that first generation, first generation, note that, hey, you could get a big H from this allele and that allele. You could get a big H, little h from either of the big H's and either of the little h's, or you could get little h, little h. You could get all three genotypes. Now, why do you get 3/4 to 1/4? Nah, you'll see in the next lesson. And we want to summarize like Mendel's four big principles, the four big things that he kind of sort of gave us and laid the groundwork here. So first one should be a common sense type thing for us. And he called it unit characteristics. The idea that a particular trait, anything you're looking at, can be controlled by a single pair of alleles. Right? Now this should make sense because you're getting one set of genetic information from an egg cell, one set of genetic information from a sperm cell. So you should have two sets of genetic information, two bits of genetic information for each trait that you've got. Right? You got one set of information from mom, one set of information from dad. Next, hey, given any trait that you're looking at, if dominance and recessiveness are in play, then one of those alleles could mask over or hide the expression of the second allele. Thus, if you got a hybrid, if you got a heterozygote, only one allele in that pair is actually going to be expressed in the phenotype. The other one's going to be hidden. Now, the other thing that Mendel figured out, and part of the genius of Mendel is he figured this out without knowing what's happening in meiosis. But he envisioned that members of each pair of alleles are going to separate when gametes are formed. And thus, each sex cell's not only going to get half as much genetic information, but only one member of those pair of alleles. This works because of what's going to happen in anaphase, when homologous pairs of chromosomes actually separate. Thus, each sex cell is only going to get one member of each homologous pair of chromosomes. Thus, each sex cell is only going to get one allele for each trait. Now, Mendel's fourth principle, independent assortment, that's a little bit trickier. And here we have to look at multiple traits at the same time, or at least two traits at the same time. And basically it says that the separation of alleles, or one pair of alleles, I should say, does not affect the separation of a second pair of alleles. Now, to envision this, the best way to do it is to go back to meiosis, or metaphase 1 of meiosis, when those homologous chromosomes are all kind of sort of paired up in a line in the center of the cell. If we had a particular gene on this purple chromosome and its corresponding allele on this green chromosome, obviously they're going to separate. They're going to go different places when anaphase occurs. Well, yeah, no biggie. That's what happens in segregation. But remember that this chromosome pair doesn't have to line up just like that with the purple on the left and the green on the right. It could just as easily have switched places. Same thing for this puppy down here or this puppy down here. Thus, since these chromosome pairs can assort themselves independently of one another, that means the alleles might separate differently depending upon how these things were arranged. Now, this is going to be a whole lot easier to see when we look at something called dihybrid crosses. But we want to go back to this diagram. Remember this diagram, and we'll show this again when we do independent assortment in dihybrids, and it should make a little bit more sense.